morning, my dear students. Today I am going to discuss about the plasmids because basically many of the students they are asking about plasmids. You need to understand about the plasmids before going to study about the structure and function of the plasmids. What exactly the plasmids are? Where do you find the plasmids, and what is the application and natural function of the plasmids? These are all the basic main questions that will be arise from any students, so that if you are familiar with these questions, got the solution, then you can clear about the plasmids. So you need to understand about what exactly the plasmids are. The plasmids are circular double-stranded autonomous self-replicating DNA. This is naturally present in the bacteria. Right, so based on the different functions, we are going to classify plasmids into different factors, right? So once again I am telling the definition. Plasmids are double-stranded circular autonomous self-replicating DNA. That means, so that take an example of bacteria which is having two DNA. One is linear DNA, another one is circular DNA. Right, and the number of plasmids also varies from bacteria to bacteria, different strains of bacteria, right, that we generally call that copy number, right? So what is the main function of plasmids? See, as I told the definition, right, it is a circular double-stranded, it is a circular DNA made by double strand, self-replicating. It has a power, right, or capacity to undergo self-replication process, and autonomous. It is not mainly depend upon the main DNA or linear DNA, right? It can function individually, right? So this is the main function. Okay, then what exactly it functions in different organisms, right? So based on the function, we are going to classify plasmids into different types, right? We can see that F plasmids. In some bacteria, we can see that F plasmids. These F means fertility plasmids or fertile plasmids. They helps in sexual reproduction in bacteria. So that there are two different types of sexual reproduction in bacteria. One is transformation, another one is transduction. And also we can see that conjugation. Transformation and conjugation that you can observe in the bacteria, but transduction that you can observe in viruses, right? In this case, these plasmids plays a very important role in transferring of genetic material from donor to recipient, right? So transformation and conjugation. So because of that basic information about the transformation, that is why we are going to study, right? So that especially DNA as a genetic material experiment, that is transformation principle or Griffith experiment, right? There actually, so that there's a transformation principle, that is the main thing where the donor DNA can capable to donate its DNA to the recipient. This transfer is mainly based on the plasmids only, so that this is a fertile. If any bacteria which is having F plasmids, this helps in fertility factor like reproduction, sexual reproduction in microorganism. This is the one type. Next, we can also observe M plasmid in some other bacteria. M plasmid means metabolic what is a plasmid? Metabolism, as you know that it is of two types, anabolism and catabolism. Anabolism is synthetic process and catabolism is destructive process, like that, constructive process and destructive process. So that any bacteria which is having M plasmids, these M plasmids enhances the metabolic process of complex biomolecules, whether it can be involved in the anabolic process or synthetic process, or it can be involved in the destructive process or catalytic process. So that M plasmids which helps in metabolic process of the bacteria. This is called M plasmids. Then apart from that, we can also observe another type of plasmids, like you just go through it, R plasmids. This R plasmid, it is also a extra chromosomal DNA, right? This plays a very important role in providing the resistance against any other antigens. Any bacteria which is having R plasmids, these R plasmids, R means resistant. Okay, these plasmids provide the resistance or gives the resistance to the bacteria against viruses or any other microscopic organism, right? So these are all the very important different type of plasmids. Okay, especially what is the application of plasmids, you need to understand, especially in genetic engineering or biotechnology or recombinant DNA technology. We are going to use the recombinant plasmids, right? So these, because of the self-replicating and autonomous self-replicating property, these bacteria, these plasmids can be, so individually they undergo replication process. So what artificially we can construct the plasmids to extract some of the important bioactive molecules. These plasmids, they can extensively use it in biotechnology or genetic engineering or recombinant DNA technology. Hence, there are different types of artificially synthesized plasmids are there. Okay, like that, you just go through it. PBR 322, T23, PUC1, PUC11, PUC12, right? Call phages, like the different type of plasmids we are going to observe. Also, this is the basic information about the plasmids, where exactly it consists, right? So that, and especially in eukaryotes also, you can see that a circular DNA, that we are not considered as a plasmids. But you need to understand, even though in eukaryotes like human beings, we are going to observe two types of DNA. One is linear DNA in all the cells, which is having so linear DNA in its nucleus. But we can see circular DNA in organelles. The organelles are present inside the cell, like mitochondria and chloroplast. These are the two important organelles which are having their own individual circular double strand DNA, right? That we generally call that mitochondrial DNA and plastid DNA, right? They are going to multiply individually, right, without the information getting from the DNA. So that, but we are not considered these are our plasmids. We are considered as a DNA. But if it is present in the bacteria, this extra chromosomal autonomous self-replicating the DNA, that we generally call plasmids. Hope so you understood well. If you have any doubts, you can post. Thank you so much.